Let's take a look at 13.8. When the amino acid sequences of insulin, the protein insulin, were isolated from different organisms. I should say, when the amino acid sequences of insulin isolated from different organisms were determined, differences were noted. For example, alanine was substituted for threonine, serine for glycine, and valine for isoleucine at corresponding positions in the protein. List the single base changes that could occur in codons of the genetic code to produce these amino acid changes. Okay, so this is like our first look at like how, even before we get to our section of the course on mutagenesis and the changing DNA sequences between organisms and evolution, and how mutations can lead to substitutions of amino acids in certain proteins in different species. We're seeing this right here in our chapter on transcription. So let me just take some notes on this right here. So we're looking at 13.8, and we are saying we're looking at amino acids sequences of insulin from different organisms. So in some of these, like certain positions would have an alanine, and alanine would be substituted for threonine. Threonine. Okay, in other positions there might be a serine, and that would be substituted for glycine. And then at another position we would have valine, and that would be substituted for isoleucine. So okay, what does this mean exactly? So that means if you take insulin, the protein insulin from several different species, and let's say you look at position, let's say at position 10, some species have an alanine, and others, have a threonine. And let's look at position 27, so the 27th amino acid in insulin. I'm just making that up. I don't know if 27 is a serine. That would be amazing. 1 in 20 chance at least, right? So let's say the 27th position is a serine, and insulin from some organisms, and from others, it's a glycine. And let's go with position 59. Okay, position 59 in some organisms it's a valine, in others it's a isoleucine. So, if insulin, the gene for insulin probably evolved one time, right, in an ancestor of, all organisms that have the insulin gene. And if that's the gene, well, mutations could have happened as, organisms were evolving. And so now we have an insulin that, has an, alanine at position 10. And then we have a version of insulin in other organisms that has a threonine at position 10. So this is insulin. So these are still the genes, and this version of the insulin gene codes for an alanine at position 10, and this version of the insulin gene down here codes for a threonine at position 10. And let's say, I don't know, let's say these are humans up here, and these are chimpanzees down here. I don't know if that's true, but that could happen. Maybe originally it was always, this insulin always coded, the ancestral insulin gene always coded for an alanine at position 10. Some mutation occurred, and now all the organisms that down here, okay, they all have a threonine at position 10, and all the organisms up here, including humans, have an alanine at position 10. That's one possibility. So if that's the case, we should be able to trace this back to the DNA and look at, what kind of changes could lead to a switch from, alanine to threonine. Let's take a look. So I'm going to cut all this out right here, and we're going to look at the genetic code here. I'm going to paste that in. Okay, so the question is, list the single base changes that could occur in codons of the genetic code to produce these amino acid changes. Okay, so let's take a look at alanine. How about, let's use red. Okay, I got my pen, got red selected. Alanine is found right here. Okay, so all of these codons code for alanine. Now let's look at threonine. Okay, where's threonine? Not the easiest. Oh, here we go. All these are threonine. So if we had our DNA, in our DNA, in the insulin gene, we had a codon that was ACU. I'll draw the other strand too of the DNA. We have TG, not U here. There would be I'm sorry, that would be, this is DNA, so this would be a T. TGA. Okay, now this codon here, that's our codon, and that's coding for threonine. Now, if there was a mutation, what would we need to mutate? What base would we need to mutate to change this threonine to an alanine? We could mutate this one right here. We could change that to a G. So let's see. So a mutation happens, that A becomes a G. So now the DNA is GCT there, and CGA here. So we have one mutation here. You color that, put that in a different color. One mutation, and now, you know, okay, this DNA is, you know, much longer than one codon, right? But when this DNA is transcribed into a messenger RNA, this one up here at that position is going to have an ACU codon, and this one down here is going to have a GCU codon. And when that messenger RNA is translated into a protein, well, this codon is going to specify a threonine to be incorporated at whatever position we said. I think we said it was position 10, but this one would say put a threonine in, and this one would say put an alanine in, GCU, right? Yep, alanine. Okay, so one base change. One base change. One mutation changes one amino acid in the insulin protein. Okay, so they're pretty much all going to be like that. I think it's all just practice of looking through the code and seeing how you can change different bases. Maybe sometimes you need to change more than one base to go from a serine to a glycine, or a valine to an isoleucine. Maybe you need to change 3 bases. So essentially this problem is just practice looking at how changes in bases and genes can lead to changes in the amino acids in the corresponding proteins. So I won't actually do the other two. I think you can figure that out on your own.